this is not a formal presentation. This is just kind of an informal presentation of some of the stuff that we were presenting at the ISNR and in other forums that Genardi and Dave Cantor and I were reviewing and talking about and then sharing with everybody. Looking at, pretty much everything we could find on neural networks. It was, what do we know about this stuff and how much of it is applicable to what we're doing with qEEG and neurofeedback? And that's an open question. Nobody has the final answer on that. You're going to have to make up your own mind, as with most of science, hopefully. But we're just trying to present the information and present some of the conundrums that this presents, but also kind of expands your vocabulary, makes you more aware of what's going on in neuroscience, what could be coming down the pike in neurofeedback. So it's just kind of a useful exercise. And so during the last couple of days of the year, we thought we'd just kind of engage in this a little bit. And if you weren't here last time, we had some pretty good discussions on these topics we were covering, and we mostly just discussed what's called graph theory, which has been a popular way to start mapping out neurons and how they're connected into networks. And what we noted was that, even though we know how neurons are connected to each other to some degree, we don't know all the dynamic details. And another issue that we have is, although we know most of all the fiber tracks coming off of the neurons in the brain now with something called DTI, diffusion tensor imaging, we don't really know how the networks emerge from all that. So you have kind of the hardware, but we don't know how the brain software emerges from all that, and that's our big problem. Or it's one of our big problems. I'm just going to see if I can put this down. Another of the big problems is the connectivity levels. We said the brain has roughly 3 identifiable levels of connections and network activity: the micro, the meso, and the macro. And it's kind of a arbitrary designation, but we've got to draw real estate lines somewhere. And as a consequence of that, we've gone into micro, macro, and meso. And we also looked at those connections, how many there were, just millions and millions of fibers, 10 to the 10 connections, maybe more, and that they are all connected at all these levels. And with different technologies, you could divide up the real estate different ways: MRI, EEG, MEG. You could define how to divide up all of these pathways into different areas. A popular one is Broadman, but there are other ways of doing it too, and that changes the picture quite a bit. And so everything is, that level of connectivity is confounding, because we can only look at one level with EEG for the most part, and that's the meso and the macro, and mostly the macro because it's not very specific. And then we discussed the fact that because of this, regions of interest have weak identity, areas like Broadman areas. In other words, they're not as well defined as we think. The cellular architecture is kind of uniform, but in terms of how the pathways through those areas go and the different nodes and hubs that those pathways hook up to are quite diverse, diffuse, and extensive. And so any given area may be related to many functions. So a lot of the connectivity varies also with what task you're performing when you're imaging somebody. What the patient group is. Turn that off. What the patient group you are using, children, adults, athletes, baseline conditions, what status they were when you started. All this affects the way you image them and can make it very elusive process identifying a region of interest, you know. The way people throw the word around, it sounds like, oh, it's an established thing. You just have to find it. You know, that's the spot for smell. That's the spot for touch. That's the spot for this task or that task. I think we're going to see it's a little more complicated than that. So we can hook the brain together with all these fiber networks. We're not sure how the software emerges from it. And we have another problem. There's a chemical connection going on all the time that we're not taking into account. So there's a lot of activity that's not being seen related to different tasks that are chemically related, neurotransmitters, and there are literally potentially hundreds of those, and many, the majority of which we've never identified. And we're still finding that neurotransmitters can make really profound changes in how they tune or sensitize a neuron to different frequencies. So all these neurons are talking to each other, but they're also sensitive to frequencies that are occurring in the interstitial fluid around them. And there's a lot of noise in the brain with all this activity. It's like being at a basketball game and sitting on the bleachers, and there's a lot of voices and noises going around you, and you can tune into different ones, different conversations around you, depending on what your interest is. And a neuron, depending on what its interest is, and its function, it can tune into different conversations passing by it as well and use that information. So that's volume conduction. And that can involve harmonics. And we can't get too into depth here, but Martin and I have talked about it a lot before. You know, if you're running something at 5 or 10 hertz, then you have multiples of that number where there's a harmonic response, a resonance that occurs. And so people like Walter Freeman, one key people who in the field who makes comments on this, has said, you know, we may be dealing with basically just a giant harmonic field, and that all the neurons are constantly tuning and communicating with each other through that field, which would change our whole concept of brain interface, actually. And in support of that, you know, we have a little document here from Meehan and Bresler, who we quote quite often, did one of the best summaries of all of this. But they're looking at a lot of the work of Bisaki, who wrote a whole book, Rhythms of the Brain, about this topic. Very interesting book, if you're interested in that, Rhythms of the Brain by Bisaki. And he did a lot of research with mice, and you can see here there's a little mouse here they're using in this picture. And what Bisaki did is he watched theta waves and gamma waves as mice ran mazes, and he began to find that there was a code here, a neural code, in which the theta and the gamma waves worked together to identify how many steps the mouse would take and what happened at each location and what it smelled, and it would all integrate that. It did it through the phase relationship between these very fast gamma waves, 38, 39, 40 cycles a second, and the theta waves, 4, 5, 6, 7 cycles a second. And this is a picture of how gamma waves occur, you know, at the same time as theta waves, and that the relationship between them, there's a comodulation process here, which we use similar process for AM and FM radio waves, where one signal carries another signal. And we could spend a whole day just talking about this topic, trying to explain it, but just suffice to know that one signal can carry another signal, and that between the phase relationship between the two, how much one is going up and down in time compared to how another one's going up and down in time, that difference can create quite a complex output, these kind of wave interferences. And that provides a way of carrying information through the brain. So the limbic system can communicate with the cortex through these gamma frequencies. And what we have is a harmonic communication. That can take place through volume conduction or through pathways. an interesting possibility. So I'd like to just bring this up, which is very important for our understanding and not too hard to grasp. It says, evidence suggests that phase synchronization of oscillations in the beta range is more important than of gamma oscillations for the interregional binding that takes place in neurocognitive networks. Those of us in neurofeedback need to pay attention to what Meehan and Braziler are telling us here. Okay, so everybody says, "Oh, I'm going to train gamma, and gamma is so important." Well, gamma is important, but it's actually it's beta frequencies that are more important for interregional binding of cognitive networks. So when you're using your cognitive networks, it's beta. Okay, and the phase synchronization of beta oscillations between temporal and parietal cortices has been associated with long-distance binding in multimodal perception. So when you're binding together a lot of perceptual processes, it's these kind of beta oscillations in the temporal beta area, and we'll talk about that farther as we get down the road. There's a couple of networks. The MTL network is part of that network, and it's a critical network for processing perception. And people with perceptual processing problems, like the problems you get, people who are oversensitive to perceptual input, sensory integration issues, are often because there's an issue in this network. And you'll see it, for instance, 70 to 75% of the people that we looked at from a statistical analysis had too much coherence in the beta frequencies in the parietal region. Well, this helps to explain why. So knowing about the network sometimes can help explain why, may not be able to fix it. And that's the theme I'm going to keep pointing at. You can often explain things or detect things, but that doesn't mean you can fix them using the same, going in the reverse of what you're seeing. The training in the parietal region might not always fix the issue. And that's a false assumption that people constantly make. So phase synchronization binds those areas. Furthermore, one study found that activity in the gamma range did not persist after driving input was removed, whereas a switch to beta oscillations allowed activity to persist in the absence of the driving input. This persistent beta activity may represent a trace of the input that serves as a kind of working memory. Okay, that's very important. That's saying that the beta is part of a process of catalyzing interfrequency activity, of carrying information from one frequency domain to the other, from theta into beta. And for us in neurofeedback, that's an important concept that we could operationalize. In vitro studies suggest that the switch from gamma to beta activity occurs as an interaction between gamma oscillations in superficial neocortical layers and beta oscillations in deep layers, such that the period of the two frequencies can contates to produce beta 14 to 20 hertz oscillations. So what we're getting is theta, subcortical waves, limbic structures, stimulating activity in the higher cortical layers, which activates deeper cortical layers in the beta frequencies. I mean, that's a banquet for neurofeedback people right there, that finding. It's why we cull through this research, because it tells us a lot about what's going on. Number one, that when we're seeing 14 or 15 to 20 hertz activity cortically, that is one of these metastable networks, these trace memory networks that last for 100 seconds or longer, and they can go on for hours, apparently. Those are our memory networks and our executive processing networks taking place. And those activities are not dependent on gamma to endure. They're only dependent on gamma to emerge. And the gamma, as far as we can tell so far, is dependent quite a bit on the theta frequencies. Okay, and there's other things we have to consider, like thalamic cortical loops, but we're not going to add that confusion here. This is just a really important thing to think about. So, yeah, you do have to have gamma. And Nunez actually wrote about this in 1995 quite extensively in his book on neocortical dynamics, but 99% of the planet wasn't paying attention and didn't know what it meant. But it was there. And I remember reading about it, talking about Jay Gunkelman about it back in the late 90s. So this is not new to us, all of us in the field, but the majority of the people it is. And at that point, I think there's only a few of us who are really looking at it saying this is important to neurofeedback. So the switch from gamma to the beta is important because it tells us that the beta frequencies are the frequencies of cognitive processing. And when you worry, so now we talk about psychology, when you worry, you're going to produce more gamma, but you're going to produce a lot more beta. Your beta is going to be ongoing. The gamma is going to be frequent and persistent, but the beta is going to be constant and ongoing. And that kind of helps support the notion that somebody with upset limbic structures, unstable affect, is going to be irritating the brain with this gamma activity, which is stimulating what we would call a lot of cognitive processing worry. And that this worry, if it goes on and on and on, is going to demand a lot of resources, a lot of glucose and oxygen and neurotransmitter, and it's going to demand a lot of clearing out too. At night when we sleep, we have to clear out all the glutamate from this activity. If we don't, it starts to generate an inflammatory response, and that reroutes serotonin to quinolinic acid, and then we start to develop depression. The other thing is that when the brain is very active in beta, Joseph Ledoux from his research in the 90s tells us that it begins to block inputs from vesicular pathways in other areas into the orbital frontal region where we make decisions. So, where we notice our worrying. So this activity can also act, if it's enough activity, to block our experience of fear from our limbic structures. This is one of the reasons, not the only, but one of the reasons that we say that if you do not have stable affect, you cannot have an efficient cognitive function. So that is a lot that you can take away from just this key finding. Just amazing where this is going here. Anybody have a question or comment on that? When I say if you look at a map of somebody with anxiety, you may not see beta. They used to say, oh, you always see beta. No, you may not. It may be physiological anxiety. They may be somaticizing it and be experienced in that way more than cortically. People do that. Most animals who are distressed highly die of gastrointestinal lesions, not as from suicide or worry like we do. So from that perspective, we can really see much more clearly what's going on psychophysiologically and how to intervene with it. And we can understand that people may have beta, or they may not have beta, and that the longer they worry, the more likely the beta is to show up, because it's creating enduring activity. And these networks are enduring longer and overlapping, and slowly the brain grows to sustain that activity. So we grow a worried brain, and then we have beta. But prior to that, from what we've seen in the maps and the analysis and what I've reported and published, is that you're mostly likely to see beta asymmetry and alpha deficits, and more likely beta asymmetry initially when people are getting fearful and worrying a lot. The alpha deficit may or may not show up depending upon how much of the cortical process has been recruited. Because if we think about it, the more the cortical process is recruited, the less alpha we're going to see, because we're going to see less evidence of the brain inhibiting areas, and we're going to have more areas mobilized for worry.
Dr. Soutar?
Yeah.
I'm curious about how coherence plays into all of this. I've worked a lot with Asperger's kids and even adults who seem to have an obsessive-compulsive phenomenology, but they also have really high coherence. Would there be more concern about coherence in one particular range? Would it be in the beta range, high beta range, that you'd look for obsessive-compulsive phenomena?
Well, that's a good question, and I haven't seen anything written about that, Carl. How coherence plays into this would be very complex, because you'd have, if you had areas that had too much demand on them, they might call upon other areas of the brain to assist. So let's say you had one region, the anterior cingulate, that was heating up a lot and was producing a lot of activity in the orbital frontal region, and that orbital frontal anterior cingulate group, which is one of the networks, FP1, FZ, was really heating up. You may have an area that is producing a lot of glutamate, right? And what if that area, what if it's particularly on the left side, because it's worry? That area might be overwhelmed, and so it might start pulling from the right side, right hemisphere. And so now you have both areas heating up, and then you'd have increased coherence measures on your map. I mean, as far as I can see in the literature, that's really what we're looking at. Resource allocation when we look at coherence. One area's pulling resources from another area in order to manage a process. I think that's about as far as I could go with it.
No, thanks. I'm really, it's interesting. I remember in my master's program in counseling, they made a big deal about saying some people are more compulsive, and those are the sensors. They're doing all the time. And some people are more obsessive. They're thinking all the time, and they're thinking that maybe it's not quite right. And usually, very bright people tend to be more obsessive, and more everyday people tend to be more compulsive. But we have these DSM-5 now categories that may not be provable when we look at the actual neuronal correlates, They
aren't. I mean, that's been very obvious they aren't. And if you think about it, the perceptual, the doing part and the motor part tends to be more posterior. And that's why you see children react more physiologically. They're more impulsive in their behavior when they're anxious. Whereas adults tend to inhibit that function as they gain frontal cortical control. But they tend to activate more in terms of social strategies with frontal development, and so they tend to be more activated in the front. And if you'll notice, a lot of the kids, if you're comparing Beck inventories like I did for years with maps, you notice the kids tend to be more activated in the back and the adults in the front. Well, guess what? In the last 6 years or so, there's been a couple studies that's published observations along that exact line, which is one of the advantages of doing Q. We see a lot of this stuff in advance. So that's an interesting way to think about what you just said, Carl, and characterize it.
Yeah, I don't want to be fooled by DSM that's been harangued. That's what the problem with DSM-5 was. They had no input from neuroscientists, none.
Yeah, and they got in trouble for that too, didn't they? Definitely.
Richard,
yes.
There was one of the honcho researchers at NIH 10, 15 years ago was talking a lot about kindling, and the psychiatrists were all parroting that. and talking about an overkindled, chronically depressed brain.
Mm-hmm.
That concept seems to apply when you're
It does.
talking about sort of the left hemisphere recruiting the right hemisphere, blah, blah,
Mm-hmm.
blah.
Yeah.
And it's usually a term from epilepsy.
Yes, it is, isn't it? But it does seem to apply. It does seem to apply, but in a more complex way than perhaps they were imagining. But it does.
Yeah,
But the original concept in psychiatry of disorder being a split between cortical and limbic function is actually having a revival right now. It really seems to be a strong theory. It's very interesting. It explains so much. Okay, so getting through that, yeah, and this will be more than just 2 sessions. We'll just keep doing this all year. We'll eventually get through it all. There's like 160 slides, and that was modest compared to what Dick had.
More bad news, Richard. I'm remembering something vaguely from the graduate school learning class that we have to hear things repeated at least 7 times before they get consolidated.
I know I have
a few more years.
I have to read an article many times before I make sense of it. People say, well, these research articles are too hard to read. I said, like, you got to read them like 7, 8, 9 times before you start to understand them. They're like haiku, you know. Okay, so let's start talking structure and function. We've been talking mostly structure, and I was getting in beyond structure with that idea that not only are things connected through defined fascicular pathways and fiber pathways, but there's also shortcuts and other pathways which are harmonic, which are electromagnetic in nature, volumetric in nature, and then there's chemical. And we don't even get that with EEG. So, let's begin to explore structure and function and how they come together. Functional nodes were first determined by lesion studies, then fMRI localization perspective. This is a lot of Dick stuff, so I'm going to interpret it. Discrete regions showing elevated metabolic activity in PET, blood flow in fMRI, spectral power in LFP, ECOG, and MEG, magnetic electroencephalography, or elevated firing rate in single unit or multi-unit recording in correlation with performance of a cognitive function. Translation: we took some piece of equipment, we looked at the brain, we performed a task, and we saw what area activated, lit up. And that is very hot. That was very hot in the 90s, and Corey Hammond and I got together and connected the MRI, the Brodmann areas, with the EEG 1020 system, and we got it from the Key Institute. And then I put all the research I could find existing at that time into a chart on what areas lit up on the MRI when somebody performed a task. And if you look at our map system, if people say, well, some of those terms are strange on the map system on the dashboard. Well, the reason they're strange is because they're all straight from the MRI research. I did not change them, although I may in the future.
Richard?
Yes.
Martin, quickly. Sorry to interrupt. Jay Gattis asked to be unmuted.
Yeah, happy.
He would like to ask questions if you can unmute him.
Thanks,
Sorry,
Martin. Yeah, hey, Jay, chime in, please.
Hi, Richard. I'm sorry, I'm a little late because we were on the beta slide when I had a question.
Oh, well, go ahead. We're not far.
You were talking about how if you are in beta and worry and then you don't recover at night, then you get inflammatory processes. Do you suspect that that's where you begin developing spindling beta?
No, that's a really interesting idea. It could be. And I don't, I've never looked into it deeply, so I'd want to look at the neurotransmitter literature on that. I would think would be the place to look at it, because it's the glutamate buildup that's causing the destabilization. But the spindling may be how that's affecting sodium or potassium pumps, or there's a lot of ways it could be affecting output. But that's an interesting thought, Jay. I have to go back to my books on that one. Well, one of the things that I was getting at by saying, well, we found these nodes, and so I put them out there, and then everybody in neurofeedback and his brother said, send me a copy. And then we got into these really exotic versions of this where they looked at every, for the next 10 years, people started mapping every single tiny task and how it was related to an activation in a particular node. This is useful, and it's useful for assessment, but we have to be careful of electronic phrenology, okay? This is the key point, because these nodes light up, and some of them are actually hubs, but they are all fed by very dispersed, diffuse network streams that merge into them. We'll talk more about that. So why they're lighting up may not be the same as that they are lighting up. So the function, that's lighting up. They're having trouble with reading, and the gyrus associated with reading comprehension is lighting up, and you're saying, oh, okay, well, that makes sense. But why is that area lighting up? Well, it's got inputs and outputs everywhere with feedback loops. You need to look at the whole field of activity to know why, and we'll keep coming back to this, and where you might intervene. And we'll keep building that theme, and the theme of the fact that network complexity, that you may have a hub or a node, or you may be able to identify 6 or 10 of them, but what about the hundreds, perhaps thousands of other feeding into them? And how are they behaving? So structural networks provide a complex architecture that provides the dynamic interaction between nodes that give rise to functional networks. Network perspective. Okay. Changes in spectral coherence between distributed neuronal assemblies may occur without changes in spectral power. That was something that Dick Genardi pulled out of all this literature and just put into one sentence, but is a very important sentence. And that is that the idea that even though these areas are lighting up, they may not be telling us about what other areas they're coherent with or discoherent with. Okay, so just the lighting up of them is not enough, that we have to look at the phase relationships between them. Important. Which I think Bob Thatcher is probably the person who's most championed that cause and delineated it, described it, and explained it with his 2-compartmental theory and all his other theories. He's done a lot of the lead work in that area. Okay, so another thing about networks and function and structure. Studies examining the total sum of afferent and efferent connections going to and from hubs and nodes, possibly, because we don't have a clear definition of hub and node. Everybody has a slightly different one. The connections going to and coming from them suggested that some cortical hub regions maintain an unequal balance of incoming and outgoing projections. So this suggests a potential role for cortical hub regions as neural communication sources and sinks. Well, this is not just a suggestion. There are a lot of people running with this idea in neuroimaging. So a hub lights up, on your brain map or whatever, whether it's MRI, EEG, or MEG, and that area may be an area that mostly puts out information or receives information. If it's a hub that's receiving more information than putting out, it may be kind of a sink, a reactive hub. And you might want to stimulate a source node or a source hub rather than a hub that's mostly a receptive hub. So that's a potential possibility. So you have to identify the hub and then whether it's one or the other if you wanted to be most effective, perhaps, an important consideration if you're maybe doing neurofeedbacks. Or maybe it's something that we're ignoring and not seeing that's interfering with what we're doing. But it's another indicator of just because an area is dysregulated doesn't necessarily mean you'll fix the functional attribute related to it, like working memory or reading comprehension. You might not fix that attribute just by training at that location. So we had people who were, using those Broadman areas and saying, oh, well, they've got working memory area, and that hub lit up the most in the literature, so I'm going to train at that area. And sometimes it would work, and sometimes it wouldn't, and they didn't know why. Well, now we know it's because of these distributed nature of networks. It's more distributed and less modular than people were imagining it. Here's some examples of these kinds of different hubs and projections. The posterior cingulate, okay, that's at around PZ. The precuneus, that's just below it. The medial prefrontal cortex, maybe up around FP1, are driven hubs or sinks. The dorsal frontal, or maybe around prefrontal, maybe that around F3, F4, posterior parietal, well, we just discussed that. Visual and insular above T3, T4, hubs are driving hubs or sources. So you can see the back is driven a lot, except for the prefrontal, the medial prefrontal. And the frontal is more of a source, and the insular is more of a source. That matches a lot of our concepts of what we were just talking about, the back being active for perceptual primary processing and related more to earlier developmental in the brain's development, and the frontal being more proactive and driving behavior, being more social and later development. So the manifestation of those networks as being driven or sources does kind of start to match up with developmental perspectives. All right, no questions or comments. So something to take away is, do we drive an area or not? A lot of times working frontally can really drive changes. But if the posterior is out, you might be spinning your wheels. All right, then there's the concept of how do we identify these locations, regions of interest, and intrinsic connected networks, the large-scale networks, the bigger ones, the more macro ones. How do we identify them? Do we identify them as aggregates of Broadman areas? Do we identify them as coordinate areas on the MRI? There's different ways to look at this. One is the popular ways is seed regions, and we've just been kind of talking about that. So based on functional interdependent analysis, you find an MRI region associated with cognitive function, or maybe a couple of them or group. They get active on the MRI. Then a map is constructed of brain voxels, all the small areas in the map, in the MRI atlas, showing significant functional connectivity with the seed region. So you look for all the other areas that seem to be coherent or coactive with respect, significantly coactive with respect to each other and to that key spot. So you kind of say, well, maybe we can, if we did that, we could figure out how the brain networks are. So you see, they don't have them figured out. This is what they've been doing for the last 8-10 years. They're saying, well, maybe this is a way to figure them out. And here's your Some ideas. Here are some seeds. So these are all names of the key areas are listed up here. And these down here are hippocampal regions. And there's our hippocampal regions there too. And all the areas around them are the areas that are feeding into them. So this is meso-level complexity. And so just kind of tune in a little farther so you can see how that works. And you can see these are actually maps. These are connectivity maps saying, okay, I'm interested in this seed region. These are all the connections, and this is how it maps. And I think that this is like looking at the Grand Canyon. It's a good idea to look at it to see this is how big solving the network problem is. This is how complex, you know. If you're feeling a little overwhelmed, that's good. That's called respect for your own ignorance and all of ours, and for the complexity of the human brain. So these are one way of constructing networks. And so this is a really agonizingly slow and detailed process. Here's one way of looking at some of these hubs or nodes. Rather than looking at it that way, we could look at it in terms of Brodmann areas. A little more comforting because they're big and they're easy, and you can actually locate them with a Loretta or even with your sensor to some degree. So we look at the precuneus, and you can see there's the Brodmann areas associated with the precuneus network. The anterior cingulate network. There you can see 25, 24, 32, 33, 10, the key Brodmann areas associated with the anterior cingulate, although that disregards the subcortical networks and any affiliated brainstem networks, which is a major problem when talking about the anterior cingulate, since those are critical frequencies, delta and theta, that help orient us towards something with our anterior cingulate. The posterior cingulate cortex, parietal cingulate cortex, you know, P3, P4. Here's some of the Brodmann areas. But again, these are general. You know, they're not like these. These are much more general descriptions of what those networks are. So these are at a much lower, you know, it's much finer scale. And these are at a much higher scale. You know, this is more macro here. We're looking at the Brodmann areas. All right, I know we're getting about done with time. So just to say in summary, components and networks cannot yet be identified at the micro level. It's too complex. We haven't been able to sort it out with all of this work. You can see some of the seed stuff is leaning in that direction. We are generally training at the meso level electrically and macro level conceptually with neurofeedback. We're training scale-free networks, which means that they organize themselves according to the same patterns, whether we're down at the micro, meso, or macro level. And we're training the major nodes from the network perspective. You know, we're training at EEG locations, C3, C4, and so forth. We're really tuning into large-scale activity that way. Okay, so we'll wrap it up for today with that, and then we'll get a little bit more into Brodmann areas and how efficient is it to train a Brodmann area, and how does that relate to fiber pathways and networks next time.
Would that be Wednesday, Richard? Wednesday, what is that, January 3rd, I believe?
It could possibly be Wednesday. We'll get with Rob and have him. I have to double check what his schedule is, and we'll see where we can fit it in next, but we'll let you know when the next one is.
Yeah, well, you know, it's anyway. Next year, Richard. Next year.
A little bit of this can go a long way. I mean, it's a lot to think about. We could maybe do one a month for a year or something, or every 2 weeks. It's up to what everybody's interested. I don't know how much ongoing interest there is in this. So if you are interested in what we've been covering and you'd like to see more of it, just let us know and we can do that. And we're going to have Dave Cantor come and talk a little bit about it too. So There'll be a lot of it this coming year, I think. All right. So with that in mind, we are recording these. We will post them up on the YouTube. I'll do it. We'll probably keep them up there for about a year, and then we'll redo them again as we learn new stuff. This changes fast, so if you're interested, as time goes by, you'll want to go up and look at them, because by the end of the year, we'll be taking them down and putting up new ones with new information. All right, we'll have a great New Year's Eve and New Year's Day. And again, we won't be meeting on Monday night because that's part of the holiday. And we'll get together on Wednesday next week. Hope you all have a great year this coming year.
Thanks a bunch.
Thank you, everybody, for being so loyal to us, to the Lunch and Learns, and I'm wishing you a very good transition into a spectacular 2018.
Yep.
Thank you.
Yep. Okay, take care.